Hello guys, in this topic we are going to discuss about safe CSOM versus unsafe CSOM. It is a topic from ENT. Right, most of the time people generally or students generally tend to memorize this particular topic, but I will suggest that you don't try to memorize this topic. Try to understand the differences between these two types of CSM in a logical manner. Unless and otherwise, what will happen that you will confuse between two options in examination and get it wrong if you try to memorize, right? So let me explain in a very logical manner. First of all, I am drawing you the anatomy of the middle ear cavity, right? This is the anatomy of middle ear cavity. What I am drawn, this particular part is not your ear pinna. This particular part is mastoid antrum, right? Mastoid space, right? This is eustachian tube, and this is middle ear cavity, right? Very easy. This is middle ear cavity. Where will your tympanic membrane be present? The tympanic membrane is present, or it consists the lateral boundary of the middle ear cavity. So your middle ear cavity, so your tympanic membrane will be somewhere here, right? So your tympanic membrane will be somewhere here. Got it? Very important. There is a space in the middle ear cavity that is present above the tympanic membrane. This space is called attic. मीन्स अगर एक हम लोग घर सोचे तो घर का सबसे ऊपर वाला जो स्पेस होता है जिसमें फैन रहता है, दैट इज कॉल्ड द एटिक पार्ट. नाउ दिस टिम्पैनिक मेम्ब्रेन होल लैटरल स्पेस को नहीं बनाता है. द टिम्पैनिक मेम्ब्रेन डजंट फॉर्म द होल ऑफ द लैटरल बाउंड्री ऑफ द मिडिल ईयर कैविटी. इट फॉर्म्स ओनली approx 80% of the lateral border of the middle ear cavity. The upper 20 part is left. The upper 20 part is actually bone, and that bone is called scutum. Exactly. So behind that scutum, that area is called attic part of the middle ear cavity, right? What is this? Mastoid space, and there is a very big cell over here called mastoid antrum. So this is called antrum or antral part, right? Now this space जहाँ पे tympanic membrane है, the space behind the tympanic membrane is called tympanic part of the middle cavity, and this space, this whole space is called tubo. Why? Because it is the space where the eustachian tube is present. This whole space is called tubo. Now we have divided this whole cavity into two parts. One which I have drawn in red, right? This particular area. This is atticoantral, right? This space contains two things, attic and antrum, so we will call it atticoantral part. And this space which I have drawn in green, right, which I have drawn in green, is tubotympanic, because it contains the tube and the tympanic part of the middle ear cavity. By tympanic part I mean the space behind the tympanic membrane. So CSOM can occur in both of these spaces, atticoantral CSOM and tubotympanic CSOM. In both of these locations CSOM can occur. Now starting with the differences between the two. Now I'm coming to the differences. Okay, first of all, अगर tubotympanic part में CSOM होगा तो कैसा होगा? What will happen if tubotympanic CSOM will occur? First of all, the tubotympanic part of the middle ear cavity is actually formed of mucosal cells. Mucosal cells. The lining is mucosal epithelium, means cuboidal epithelium, right? It is cuboidal epithelium. So if mucosal or cuboidal epithelium is present, जो secretion होगा, the secretions will be mucoid secretions, right? Mucoid. Since these cells are actually specialized for secreting mucus, that's why the secretions will be profuse, because they are mucoid cells. If the secretions are profuse, they will also be non-smelly. Always remember, profuse secretions are non-smelly. Also mucus is non-smelly substance. Okay, they are not very heavily smelly. Also, since it is mucus, it will be non-purulent, right? Because mucus contain antimicrobial actions, right? So it will be non-purulent. Okay, so it is, the thing I've told you is about secretions. Now next thing, will blood be present in this type of secretion? Blood will be present or not? Answer is no. No blood. Why? I will tell you later. Now coming to atticoantral part. Now this particular atticoantral area of the middle ear cavity is actually formed of squamous epithelium. You all know that it is formed of squamous epithelium. Since it is formed of squamous epithelium, so since squamous cells are not designed or not actually made to produce secretions, that's why what will happen, the secretion will be scanty. बहुत कम रहेगा, secretion will be scanty. Plus secretions will be smelly, because the secretions are scanty, secretion will be smelly, plus it will be purulent also, right? The secretions will be purulent also. Will blood be present? The answer is yes. Why blood will be present? Because squamous epithelium is a very thin structure. You all know that when squamous epithelium gets eroded, or CSOM occurs out of squamous epithelium, this particular thin epithelium will get eroded very easily, and if it gets eroded, the underlying actually the part will get exposed, which will have blood vessels, right? And so blood will actually come out of this. So blood will be present. It is plus minus, blood may or may not be present. But in tubotympanic part, it is mucoid, right? The cells are very big, it is cuboidal. That's why it is difficult to erode, and you will never get blood in it. Okay, so what do you think, whom do you think is actually safe and unsafe among them? Of course, blood is coming out of it, aticoantral, so we can call it unsafe, right? Unsafe CSM, right? And since tubotympanic there is no blood, we can call it safe. Now the interesting part is that we don't call them safe and unsafe based on these differences, right? The main difference is actually granulation tissue. In tubotympanic part there is no granulation tissue. The granulation tissue formation is absent, right? Since there will be no granulation tissue, there will be no cholesteatoma, right? And since there will be no cholesteatoma, there will be no complications. Whereas what are the complications of cholesteatoma? There are many, like nerve impingement, bone erosion. These are the examples. Whereas in unsafe CSM, they are called unsafe not because of blood, because granulation tissue formation is present. Because of granulation tissue formation, you will be able to see cholesteatoma, which is a very dreaded complication, right? Cholesteatoma will be present, and because of cholesteatoma, complication will also be present. Because of cholesteatoma, there will be various complication. That's why it is called unsafe, not because of blood, right? So we have basically discussed everything important about this particular these two differences in a very logical manner. I think that you won't be able to forget after I have explained in this manner. So let's start the difference between these two from the table I have taken from Marrow. First of all, tubotympanic. Here I have written tubotympanic in this side, right? And here it is given in this side. Don't get confused. So tubotympanic, we all know it is a mucosal variant because the cells are mucoid epithelium. Since it is mucoid, it will have profuse secretions. It will be mucoid. It will be non-foul smelling. It will be non-blood stained because the epithelium is thick. It will be present in protympanon. What is protympanon? That area, which is that part of the medullary cavity which is present behind the tympanic membrane, right? This particular area, this thing, this from here to here, it is called protympanon, right? And this area is called epitympanum. This area is called protympanum, right? Got it? So it will be present in protympanum and also around the eustachian tube, because the name only tells you tubotympanic, right? So how much easy can it be? Very important thing we forgot to discuss, that perforation. Very important, tubotympanic. See, here is the eustachian, here is the tympanic membrane, right? So अगर tubotympanic CSOM हो रहा है, if there is tubotympanic CSOM, there are likely chances that you will get a perforation over here. Whereas if aticoantral part CSOM is occurring, aticoantral part, see, this atical part CSOM is occurring, it is only touching the one margin, so it will cut out the margin of the tympanic membrane. That's why what happens that in tubotympanic variety you will get central perforation, right? Whereas in aticoantral you will get atical or marginal perforation, because अगर यहाँ पे CSOM होगा, it cannot touch the central part, वो lateral part को ही touch करेगा. It will only touch the lateral part of the tympanic membrane, and hence it will erode that part only and cause perforation in that part only. That's why marginal or atical perforation, right? Tubotympanic, because it is safe, no granulation tissue, already told, no granulation tissue. Pale polyps are also rare, and pale cholesteatoma, that's why it is absent, and there, of course, because cholesteatoma is absent, so rare complication, so we can treat them medically or surgically, extremely easy, right? In aticoantral part, it is unsafe. We all know that. Why is it unsafe? It is unsafe because granulation tissue is common. Then granulation tissue is common, so cholesteatoma is present. If cholesteatoma is present, then complications are also present. Then if complications are present and complication occurs so fast, so only surgical procedure is done, mastectomy. We don't go for medical management in such a case. Because aticoantral part has squamous epithelium, it is also called squamous cell disease. Since it is squamous cell, it will be scanty, purulent, foul smelling, and may have blood because it is thin, and it will be present in the atical and mastoid antrum part. So we have completed this whole topic very nicely. I think that you won't be able to forget this particular topic after understanding in this manner. Thank you.